Hello, my dear students. Myself, Dr. Pitambar Humane, founder Botany for You, and working as professor and head department of botany, Dharampeth MP Dev Memorial Science College, Nagpur. First of all, I welcome you all in this online platform for the students of Life Sciences Botany for You. Friends, if you are new or not yet subscribed our YouTube channel, then kindly like, share, and subscribe our YouTube channel Botany for You. In our last lecture on question bank, we have seen the different questions asked in university examinations for B.SC semester 1 botany 1. In today's lecture, we will see the another paper, that is B.SC semester 1 botany paper 2, which is based on fungi, plant pathology, lichens, bryophytes, and mushroom cultivations. In this lecture, we will have again 4 units. First one based on the fungi, second unit based on the plant pathology and lichens, third one on the bryophyte, and fourth one on mushroom cultivation. In first unit, we will have the general characters, classification, and economic importance of fungi with 4 different fungi, that is albigo, mucor, puccinia, and cercospora in the form of life history. In second unit, we will have the plant pathology, in which we will study the 3 different plant diseases and the lichens. In third unit, that is general characteristics, classification, and economic importance of bryophytes and life history of Mertensia, Anthoceros, and Funaria. And in last unit, that is skill based unit, that is the mushroom cultivation, in which we will see the introduction of mushroom cultivation, then technology of mushroom cultivation, and techniques that is to be used for the culture or growth of the mushroom cultivation. As we have seen earlier also in last paper, that the question paper is based on the different 4 units and consists of 5 different questions, of which the question number 1 based on the unit 1, question number 2 based on the unit number 2, question number 3 on unit 3, and question number 4 on unit 4. And every question is provided with 2 5-mark questions, and in option there are 2.5-mark questions which are 4. While the 5th question is based on all 4 units, that is unit 1, 2, 3, 4, and there are total 12 notes, of which we have to attempt any 10, and 1 will be for each mark, or 1 mark for each. So in this way, the total 50 marks will be there on the question paper. As we have seen earlier, that there are the 3 different type of the questions: 5-mark question, 2.5-mark question, and 1-mark question. So step by step on every topic we will see the different type of the questions asked in the university examinations. Here unit 1, that is the fungi, and 5-mark question will be asked, that is the general characteristics of fungi. So we have to provide the all general characteristics of the fungi, that what is the cell structure, how they are producing or reproducing themselves, then what are the different diseases, plant diseases, animal diseases, etc. So all such type of the general characteristic that we have to provide in this type of the question. In second question, that is the classification of the fungi, which is provided by the Alexopoulos in 1996. Here already for your understanding, I have given the classification in a short, that is the Alexopoulos has classified the fungi into the 3 different kingdoms, that is the kingdom 1 fungi, then kingdom 2 that is the Stramnophylla, and the kingdom 3 that is the Protista. The first kingdom, that is the fungi, is classified again into the 4 different phylums, that is the Chytridiomycotina, then Zygomycotina, Ascomycotina, and Basidiomycotina. While the second kingdom, that is the Stramnophylla, is again classified into the 3 different phylums, that is the Oomycota, then Hypochytrymycota, and Labyrinthylomycota. And the third one, that is the kingdom 3rd, that is the Protista, again is classified into the 4 different phylums: Plasmodiaphragmomycota, then Dictyosteliomycota, then Acrasiomycota, and Myxomycota. So in this way, we can provide the classification which is provided by the Alexipolis. Then economic importance of fungi, in which we can provide the negative as well as the positive economic importance. The fungi are used as a food material. The fungi are also useful in different medicine preparation. Fungi are also excellent source of the vitamins and the minerals. Again, they are very commonly used in the different biological experiments, even in the genetic engineering. So such type of the negative as well as the positive economic importance that we have to provide in this type of the question. Second type of the question, that is the 2.5 mark question, that we have to provide the useful aspects of the fungi. See now, the economic importance of the fungi, we can provide the useful aspect as well as the harmful aspect, and both of these are asked in separately in 2.5 mark questions. That is for 2.5 mark useful aspect, and another question that is the harmful aspect for the 2.5 marks. Then again, the economic importance of the fungi, that earlier in last lecture we have already discussed that it is not necessary that the 5 mark question will not be asked for the 2.5 or 2.5 mark question will not be asked. Such type of the system is not there. Any type of the question can be asked for the 2.5 mark as well as the 5 mark. That will be completely depend upon the paper setters. Then classification of the fungi, outline only. Just we have to provide this outline of the fungi, and that will be sufficient for the 2.5 marks. Then general characteristics of the fungi. Again, this question was earlier asked for the 5 marks also. So again here the same question is asked for the 2.5 mark. Then in a 1 mark question, that is imperfect fungi. So imperfect fungi means what? So those fungi which are unable to reproduce themselves by means of sexual reproduction, or in other words we can say, when true sexual reproduction is absent, then the fungi are known as imperfect fungi. Then second one, Haustoria. We know the fungi is a group of the organism which is depend upon other organism for its mode of the nutrition. Means it is a group of heterotrophic organism. So when it is absorbing the food material from other organism, that is by means of sending a protuberance from the hypha, and that is known as the Haustorium. And this Haustorium is responsible for the absorption of the food material from the host plant. Then the mycelium. If you see the hypha and hyphae, so hypha is a singular word, then hyphae is a plural, and when there is a tangled mass of the hyphae, then it is known as the mycelia. So there may be 1 mark question on the mycelium also, hypha also, or hyphae also. Hypha is a vegetative part of the fungi. Then obligate parasite. Fungi, many fungi are the obligate parasite. Those fungi which can complete their life cycle only on the one host, then it is known as the obligate parasite. Then cenocytic hyphae. If you see these hyphae of the fungi, you will find that here there are the septum in between the nuclei in some of the hyphae. While if you see the another type of the hyphae, here there are not the presence of such a type of the septum in between the nuclei, and such a type of the hypha without a septum is known as the cenocytic hyphae. Then saprophytic fungi. We know the fungi are showing the different mode of the nutrition, that they are the parasitic or they are saprophytic. If you see the parasitic fungi, they are depend on other living organism causing the diseases, and in case of some other fungi, they are growing on dead and decaying organisms or the material, and that type of the fungi which are growing on the dead organic matter, that is known as the saprophytic fungi. From the second unit, the another one, that is the albugo, which is one of the genera and is responsible to cause the white rust disease. So question for the 5 mark is the sexual reproduction in albugo. If such a type of the question is there, that is the sexual reproduction, we have to provide the data regarding the structure of female reproductive organ as well as the structure of male reproductive organ. Then formation of the gametes, fusion of the gametes, and then formation of zygote. Then after formation of the zygote, there will be a meiosis and formation of the meiospore, which germinate to form the new hyphae. So this is given in the sexual reproduction. In asexual reproduction, albugo reproduces by means of the formation of conidia, and these conidia are formed on the conidiophores from the hyphae develops on the host plant. If you see this structure, it is a conidiophore, and from it there is development of many conidia. So this asexual reproduction in albugo is by means of formation of the conidiophores or conidia, and these conidia germinate to form the new hyphae and are ready to infect on the other healthy plants. Then again the same, that is the asexual reproduction in albugo, ask for the 2.5 marks. Means the same question can ask for the 5 marks also as well as the 2.5 marks. Then sex organs in albugo. The similar type of the question, that is the sexual reproduction in albugo. It is asked in some different manner, that we have to provide the structure of sex organs, that is oogonia and antheridia. So first of all, we have to provide the structure of oogonia, and then we have to provide the structure of antheridia. So that becomes now the sex organs in albugo. Then again sexual reproduction in albugo, we have to describe this structure of the male and the female reproductive organ, along with we have to provide the fertilization and the formation of the meiospores. Then one more question on the albugo will be the white rust. This is the disease which is caused by the infection by the albugo on different plants of the Amaranthacea as well as Brassicaceae convolvulaceae. So very commonly you can find here there is formation of the white patches after the infection by the albugo on the different plants or the leaves or the stem of the different plant, and such type of the white patches which are formed after the infection by the albugo, that disease is known as the white rust. Then oospore. Oospore is a name of the zygote. So if you see the female reproductive organ and the male reproductive organ, after the fertilization, here male gamete is released and formation of the zygote. Then this oosphere later on convert into the oospore, that is, it is nothing but the zygote after the fertilization. Then the next one is mucor. In a mucor, very commonly the question is asked on the sexual reproduction in mucor for the 5 marks. And in a sexual reproduction, we have to provide such a type of the data that there is a formation of gametangia from the male and the female thallus or the hyphae. Simply, they are known as the plus and the minus. The plus one is a male and minus one will be the female. So such type of the two hyphae, when come close to each other of the opposite strain, that is plus and the minus, then there is formation or the separation from the parent hyphae, and then there is formation of the progametangia and later on the gametangia. After the fusion, there is a formation of the single cell, that is the zygote, and from this cell there is formation of a zygospore. Means the total number of the nuclei which are present on one hyphae of the plus strain and the other nuclei which are present in the hyphae of the minus strain, they fuse together. That is the one nucleus from the plus strain and another nucleus from the minus strain. such type of the many zygotes are formed, and hence also it is known as the cynozygotes. So in this way, after formation of the zygospore, there is a germination of the zygospore and then formation of the zygosporangium, and inside the zygosporangium there is formation of the spores, and these spores are haploid. Then thallus structure in mucor. In thallus structure, we have to provide the data regarding the structure of hyphae and the formation of reproductive structures on it. Then zygospore formation in the mucor, again it is a sexual reproduction in mucor. Then again 2.5 mark question, that is the asexual reproduction in mucor, and the asexual reproduction in mucor is this one, that is a formation of a sporangium and spores. So this is very common method of the asexual reproduction in mucor, that from the hyphae there is a development of the sporangiophores. From the sporangiophores there is a development of the sporangia, and inside the sporangia there are the development of the many spores, and this is carried out in a favorable condition. Then again the sexual reproduction in mucor, which is asked for the 2.5 marks. Then sporangium in mucor. So again here the sporangium, that is the asexual reproduction in mucor. Then mold. Here it is a saprophytic fungi, and there is a development of the hyphae from all the sides of the braid or any other organic matter, and such a type of the fungi are known as the mold. It develops on the braid, and hence it is known as the braid mold, and spores which are formed by it, they are black in color, and hence it is the black mold also. Then saprophytic fungi. Earlier we have seen that saprophytic fungus or the fungi means the fungi which are growing on the dead organic matter, that is known as the saprophytic fungus, and the mucor growing on the braid or any other organic matter, and hence it is also a saprophytic fungus. Then columella, again 1 mark question, and columella, if you see, there is a presence of a sterile part in the center of the sporangium, and that is known as the columella. So it is responsible to provide the nourishment to the developing spores inside the sporangium. The next fungus, that is the Puccinia, and on the Puccinia there are the lot of scope to ask the questions in the examination. In a 5 mark question, we will have the acial and the pycnidial stage in Puccinia. So in a acial and the pycnidial stage, that is the acidiospores or the aciospores, and pycnidiospores or the pycnidial spores. So these two type of the spores, if you see here, this is a pycnidiospore and pycnidial cup, and this is a aciospores or acial cup. So we have to provide this diagram along with the description. Earlier already we have discussed that whenever the question is asked for the 5 mark or 2.5 mark, and if there is a diagram, you must draw the diagram, as the question is asked with a particular bifurcation or the description of the marks. And in it, if the 5 mark question is there, even there are the 2 marks or 1.5 mark on the diagram, and rest of the marks are provided on the description. And hence the diagrams are also equally important while attempting the questions in the university examinations. Second question for the 5 mark on this Puccinia is Uridosaurus and the Teletosaurus in the Puccinia. If you see this one, first uppermost diagram, that is, you will find here there is a development of the uridospores, and the uridospores develops from the structures, and that is known as the Uridosaurus. And if you see the another one, that is the teletospores, which are developing from the structure, and that is known as the Teletosaurus. So we have to provide both these diagram, that is the formation of the Uridosaurus as well as the Teletosaurus. Then the stages of Puccinia on the wheat plant. For such type of the questions, you must know that which type of the spores are formed on the wheat plant and which type of the spores are formed on the barberry plant. So on the wheat plant, there is formation of the uridospores and the teletospores. So we have to provide both these one in case of such type of the question. In Another fourth question for the 5 marks is that the stages of Puccinia on barberry plant. And if you see this, the pycnidiospores and aciospores, so these are formed on the barberry plant. So in this way, you can provide the diagram with the description according to the questions. For 2.5 marks, here there are the teletospores. So simply you have to draw this and see the difference that the teletospores are bicelled while the uridospores are binucleate. This is the very common. Again, the exine is echinate in case of the uridospores and the exine is smooth in case of the teletospores. Then teleal stage in the Puccinia, same you have to write in the question. Then acidial stage in the Puccinia, that the formation of the aciospores or the acidiospores, that becomes now the acidial stage in Puccinia. Then acidial cup in Puccinia, this structure which is given, that is known as the acidial cup in the Puccinia. Then uradium and thallium in Puccinia, the same question, that is the uridosaurus and the teletosaurus, which are earlier asked in the second question, that is for the 5 mark. So we have to provide the same type of the description. Earlier you might have read that the uridospores are formed in a favorable condition, and when there is a onset of the unfavorable condition, then there is a formation of the teletospores by the Puccinia. Then uradial stage in the Puccinia means the formation of the uridospores by the Puccinia. Then pycnidial stage of the Puccinia, so you can write about the formation of the pycnidiospores or the pycnidial cup in case of the Puccinia. In case of the 1 mark question on the Puccinia, there is formation of the uridospores. So in such type of the question, you can say that uridospores are formed inside the uridosaurus on the wheat plant. Uridospores are formed during the favorable condition, and the exine of the uridospore is echinate. So such type of 3 to 4 sentences are very sufficient in case of the 1 mark question. Then basidiospores, after the germination of the teletospores, here there is formation of the basidiospores. So the basidiospores are the haploid in nature. Then disjunctor, if you see the cell or the structure which are formed by the aciospores or the acidiospores, they are hexagonal in structure and they are binucleated. So these are known as the aciospores or the acidiospores, and each aciospore or join with another aciospores by means of the cells which are sterile in nature, and they are known as the disjunctor cell. They break to release the aciospores. Then acidium or pycnidium, this cup-like structure which is present on the ventral side of the barberry leaf, that is known as the acidium, and this cup-like structure which is present on the dorsal side of the barberry leaf, that is known as the pycnidial cup or the pycnidium. Then rust disease, Puccinia cause the disease on the wheat plant, that is known as the rust disease. So rust, that is either black rust, brown rust or the yellow rust. So you can find here, these are the different diagrams which show the rust disease which is caused by the Puccinia on the wheat plant. Then heterogeneous parasite, whenever we are talking about the parasite, they are forming or causing the diseases on the other organisms. So one organism living on the other organism and obtaining the food material. So if you see the heterogeneous, then it means that whenever the fungi or the organism complete its life cycle on the 2 different host, then it is known as the heterogeneous parasite. We know that the Puccinia use the 2 different hosts. One is wheat plant and the another one is a barberry plant. So these 2 hosts are used by the Puccinia to complete its life cycle, and hence this fungi is known as the heterogeneous parasite. Then again teletospore, earlier we have seen that the teletospores are the bicelled spores and later on they germinate to form the basidiospores. Then epibasidium and the hyphobasidium. If you see this structure of the teletospore, here this cell which is involved in the formation of the basidiospores, the lowermost in is known as hypobasidium, and this upper tube-like structure which is responsible for the development of the sterigmata for the formation of the basidiospores or release of the basidiospore, and this is known as the epispores. So this way we can say that here there are the epibasidium and the hypobasidium structures on the teletospores during its germination. In next, that is Cercospora, the 5-mark question very commonly asked, that is the sexual reproduction. If you read earlier, then you will find that Cercospora reproduces by means of only asexual reproduction, and sexual reproduction is found to be absent in the Cercospora. So always there is a question on reproduction in Cercospora or the asexual reproduction in Cercospora. It means that the answer is same, that is the asexual reproduction. And here the asexual reproduction by means of the formation of the conidia, and the very specifically here the type of the conidia which are formed by the Cercospora, they are multicellular and mostly they are falcate or the curved. Then asexual reproduction in a Cercospora, again the same question was asked for the 5 marks and the same question is asked for the 2.5 marks. Then symptoms of Ticca disease and asexual reproduction in Cercospora. So when there is a infection by the Cercospora on the groundnut plant, then there are the formation of dark brownish spot on the leaves, and this is known as the Ticca disease. So we have to provide the description regarding the symptoms of the Ticca disease and also the same description that we have given for the asexual reproduction. Then in a 1 mark there is question, that is the falcate conidia. Falcate means such a type of the sickle shaped structures, and they are multicellular, and such type of the conidia are known as the falcate conidia. Then geniculate. So again here the geniculate is used for the same, that they are curved or the sickle shape. Then knee joint. If you see the conidiophore, this is a conidiophore, and if you see the structure of the conidiophore, then you will find that it is just like to that of the knee, and hence it is known as the knee joint. Then the Ticca disease, that is the disease which is caused by the Cercospora on groundnut plant, and that is known as the Ticca disease. Then in the second unit, that is the plant pathology, here the questions for the 5 marks will be on each plant disease. Already we have seen that in the syllabus there are the 3 different plant diseases: citrus canker, red rot of sugarcane, and leaf curl of papaya. The citrus canker is caused by Xanthomonas citri. Then red rot of sugarcane is caused by Coletotrichum falcatum, and the leaf curl of papaya caused by the begomovirus. Means one is a bacterial disease, another is a fungal disease, and third one is a viral disease. So 3 different type of the diseases. So in such type of the question that may ask for the 5 mark or may ask for the 2.5 mark, what we have to write? We have to write the symptoms of the disease, then causal organism, and the last one is control measures. So these 3 points are expected if the question will be for 5 marks or even if it is asked for 2.5 marks. So same type of the questions are asked for 5 marks as well as the 2.5 mark in this type of the unit. Then in 1 mark, here question number 1, that is the host. When any pathogen infect on another organism, then that organism which is infected is known as the host organism, and the organism which infect on that host, that is known as the pathogen, or we can say that the causal organism of any disease, that is also known as the pathogen. Then symptom. If you see any disease or any plant disease, say for example, if you see this citrus or the leaf and the fruit of the citrus, you will find that here there are the development of the certain spots on the fruit as well as the leaf of the citrus. So this is the identification mark or certain morphological characteristics on the basis of which we can identify this disease, and such a type of the characteristics are known as the symptom. Then red rot of sugarcane, even it is asked for the 5 marks, asked for the 2.5 marks, and even it is asked for the 1 mark also. Then fungicide, we can use the fungicide here to control the red rot of sugarcane, as it is a fungal disease. Then leaf curl of papaya, again the another type of the same question for the 1 mark. Then Bordeaux mixture, that is again used to control the plant diseases. And the plant pathology means the branch of biology which deals with the study of the different plant diseases, that is known as the plant pathology. So in this way, 5 mark question, 2.5 mark which are the same on the plant pathology, and even the 1 mark questions are also same. Very rarely few different type of the questions are asked in this unit. Even there will be a question on the 1 mark, that is on the causal organism, means simply there may the Begomo virus will be given, and that will be for the 1 mark, so that we can even provide the same type of the answer but in a 2 to 3 lines for the 1 mark questions. In second unit, the another topic is from the lichens. Again, in a 5 mark question, there are the types of lichen, first question, and in a types of lichen, we have already studied the 3 different types of the lichen, that is the foliose lichen, fruticose lichen, and crustose lichen. So along with the diagrams or the figures, we can describe these 3 different type of the lichen in this question. Then vegetative reproduction in lichen, again the 5 mark question earlier asked in the university examination. Very commonly there is a development of the organism by means of vegetative reproduction, either by fragmentation, by means of isidia, by means of soredia, and by means of cephaloids. So cephalodeum. So here there is a formation of the isidia, formation of the soredia, and formation of the cephalodeum on the plant body, and even by means of fragmentation of the thallus there is a formation of the new organism or the independent organisms. Then VS of apothecium in lichen, this is very commonly asked question for the 5 marks as well as the 2.5 mark, and this is a diagram that is expected in this question, that is the VS of apothecium, that is the VS of the thallus along with the apothecium VS, along with here there is a formation of the ascus with the ascospores. Then VTS of the thallus of lichen, so we have to provide the VS of the thallus so that we can find here the internal structure of the thallus. Then VS of apothecium in lichen, diagram only, means in this question we have to draw this diagram only. Then apothecium, we have to describe this structure of the apothecium, which is the fruiting body. Then economic importance of lichen. Lichen are widely used for the different purposes. For example, the lichen used as a food as well as the fodder. They are used in making the dyes. They are used in preparation of the perfumes. They are used in the medicines, etc. So such type of the different economic importance that we have to give in this question. Then again, that we have seen in the earlier question, that is the types of lichen. There are the 3 type of the lichen. So each type can also be asked for 2.5 mark question. Then again, the types of lichen, see, it was earlier asked for the 5 mark question, and even it is asked for 2.5 mark question. So both type of the interconversion of the questions will be there according to its description. Then the vegetative reproduction in lichen, the same question was asked earlier for the 5 marks also. Then there will be again 1 mark questions on the lichen, that is the soredia. We have seen that it is a structure which is developing on the thalli. or the thallus of the lichen, which is used for the vegetative reproduction. Then symbiosis. Here, if you see the lichen is a symbiotic association in between the algae and fungi. Means both the organism living together with mutual contact or with each other and having the benefit of both the organism. Such type of the system of living is known as the symbiosis. Then crustose lichen, foliose lichen, fruitiose lichen, even any one of them can be asked for 1 mark in the question. Then Ecidia is a structure which is used for the vegetative reproduction. Then here the structure of rhizines, which are used for the absorption of the minerals and the water from the ground. Then cephalodia is a structure which is developing on the thallus and is also used in the vegetative reproduction. Then ascocarp. If you see the formation of the ascus in a apothecium, here there is a presence of a ascocarp, a common structure in which there are the development of the meniaci. Then medulla is a central part in the thallus which is made up of many hyphae of the fungi. Then ostiole. If you see the structure of the female reproductive organ or the male reproductive organ, very basically the male reproductive organ. So there is a presence of a pore to the terminal side, and such a type of the pore is known as the ostiole. You can see it in a diagram. Then symbiotic association. This symbiosis or the symbiotic association, both are the same here, in which the algae and fungi are living together. In third unit, we will have the general characteristics of the Bryophyta, classification of Bryophyta, along with the 3 different genus that we have seen earlier. Here, first one, that is the Prosker classification in bryophytes. The bryophytes are classified into 3 different classes, that is class 1, class 2 and class 3. The first one, that is Hepaticopsida, second one Anthoceratopsida, and third one is the Bryopsida. First one classified into the 4 orders and 9 families. Second one, that is Anthoceratopsida, classified into 1 order and 1 family, while the third one, that is the Bryopsida, classified into the 17 subclasses, 17 or 3 subclasses, 17 orders and 81 families. So in this way, if there will be the question that we have to provide such a complete data regarding the classification of the bryophytes provided by the Prosker. Then economic importance of bryophyte. Again, the bryophytes are economically important. They are indicators, ecological indicators. Even they are used for the food or the medicine, and in this way we can provide the different economic importance. Then elaters. Elaters are the hygroscopic structures found in the bryophytes in the capsules, which are involved in the spread of or dissemination of the spores when the capsule broke down. Then bryophytes as amphibians. If you see the bryophytes and its life cycle, then you will find that there is need of the water for the sexual reproduction, as the male gametes are motile and that can move from antheridia to archegonia. But if you see the structure of the bryophytes, they are provided with the rhizoids and they are growing on the soil. It means that the sexual reproduction require the water for the bryophytes, and for growth and development there is need of the soil. Means here water as well as the soil both are required to complete the life cycle of the bryophytes, and such a type of the organisms are known as the amphibians, and hence bryophytes are known as the amphibians or the amphibians of the plant kingdom. Then rhizoids, there are the root-like structures but not the root. They are developing from the ventral side of the thalli and are responsible to anchor the plant body, as well as they are involved in absorption of the water and mineral from the soil. Then gametophyte, basically the dominant phase of the bryophyte is gametophyte. which is involved in the formation of male as well as the female reproductive organ, that is Antheridia and Archigonia respectively. Last one for the 1 mark on the Bryophyta, that is the mosses. If you see the classification, that is Hepatocopsida, Anthoceratopsida and the Bryopsida. The organisms which are involved in the Bryopsida are known as the mosses. Specifically, or unique characteristics of the mosses is that they can store the 100 times water than their dry weight, and hence they are used very commonly in case of different gardening experiments. The next one in the unit, that is the Mertensia. If you see the 5-mark questions on the Mertensia, here the first question, that is the LS Antheridiophore. If you see the Archigoniophore and the Antheridiophore, so from the thallus, here there are the development of structures, and they are known as the Antheridiophores and Archigoniophores, because there is a development of Archigonia as well as the Antheridia from these structures. So in such type of the question, we have to draw the diagram of LS of Antheridiophore as well as the LS of Archigoniophore according to the question, and to provide the description also. Third question, that is LS Mertensia sporophyte. If you see the bryophytes, this is a very common question which is asked in the university examination, that is the structure of sporophyte or the capsule or the sporogonium, one and the same thing. So LS of Mertensia sporophyte means, if this type of the question, we have to draw this diagram and provide the description of the foot, seta and the capsule of the sporophyte. Then VS thallus of the Mertensia. So in case of the VS thallus of the Mertensia, the characteristic feature of the Mertensia is that there are the presence of the two different zones, that is the upper one photosynthetic or the assimilatory zone, and the second one, that is the lower one, that is storage zone. Again, there are the development of the rhizoids of the two different types, then scales of the two different types, presence of the breathing pores or the stomata which are present on the upper side are used for the exchange of the gases, having the chloroplast in the photosynthetic filaments. So these are the some characteristic features of the thallus of the Mertensia. Then in case of the 2.5 mark questions on Mertensia, that is the LS of sporophyte of Mertensia, see here only the diagram is asked, means we have to just draw this diagram for the 2.5 marks. Then VTS of the Mertensia thallus, again the same question was earlier asked for the 5 marks, and only the diagram is asked for the 2.5 marks. So this is very common questions in case of the Mertensia in the university examinations. Then Jimma cup. If you see the vegetative reproduction in the Mertensia, one of the methods of the vegetative reproduction in Mertensia is Jimma cup formation, and to form the Jimmy inside the Jimma cup, which detach from the plant body or release from the cup and grow with the help of growing notch and can form the new thallus. Then Antheridiophore of Mertensia means on which there is a development of the Antheridia, and Archigoniophore of Mertensia from which there is a development of the Archigonia or the female reproductive organs. If you see the 1 mark questions on the Mertensia, then tuberculate rhizoids. You know, in case of the Mertensia, there are the two type of the rhizoids. One is smooth walled rhizoids and second one tuberculate rhizoids. So if you see the rhizoids, there is a peg-like ingrowth in the lumen of the rhizoids, and such a type of the rhizoids are known as the tuberculate rhizoid, which is the characteristic feature of the Mertensia. Then Antheridiophore and Archigoniophore, that we have seen earlier, that there is a development of the structures like the umbrella on the thallus, and that is known as the Antheridiophore and Archigoniophore, as they are involved in the formation of these sex organs. Then Jimmy, that is vegetatively reproducing bodies which are involved in the vegetative reproduction. Then Hepatocopsida, we have seen in Prosker's classification. Hepatocopsida is one of the classes which are provided by the Prosker, that is Hepatocopsida, Anthoceratopsida and the Bryopsida. So Hepatocopsida is the class in which the Mertensia is included. Then Jimma cup is the structure which is present on the thallus of the Mertensia and is involved in the formation of the Jimmy. Then appendiculate scales, we have seen that the Mertensia thallus form the two different type of the scales on the ventral side, simple scales and the appendiculate scales. Then photosynthetic filament, if you see the VTS of the thallus, the upper zone is provided with the assimilatory or the photosynthetic filaments which are involved in the process of photosynthesis. Then areolae is a structure which are involved in the exchange of the gases and are present on the upper side or the dorsal surface of the thallus. Then there is a covering or there is a outermost cover of unicellular or one cell thick, and this type of the structure which is present around the sporophyte of the Mertensia, that is known as the calyptra. Then pseudoperianth or the peridium. So around the capsule or the sporophyte of Mertensia, there is a covering or one or two layers cells that is known as the pseudoperianth, or it is also known as the peridium. Then the next genus in the bryophyte, that is second one, that is the Anthoceros. Anthoceros, if we see the difference in case of the Mertensia and Anthoceros, you will find that the internal structure is different. That in a Mertensia there are the two zones, that is upper photosynthetic and lower one storage zone. Such type of the bifurcation of the two zones are not there in Anthoceros. So here this is a structure of internal structure of the Anthoceros thallus. The first one, that is the LS sporophyte of Anthoceros. Already we have seen that there is a very common question in case of the bryophytes on the sporophyte. Always there will be the question on the LS of sporophyte. Then second one, that is the VTS of thallus of Anthoceros. So we have to provide here this structure, that is the VTS of Anthoceros, and here this is the LS of sporophyte of the Anthoceros. That here also you will find that the foot, seta and the capsule. Then VTS of the Anthoceros thallus. Again 2.5 mark question, the same. Then VTS thallus diagram only, means we have to draw this diagram only. Then internal structure of Anthoceros thallus, again the same question. Means very commonly the question on the Anthoceros is on either thallus or on the sporophyte. Then antheridium of Anthoceros. If you see this structure of the thallus developing the antheridium, very commonly the two antheridia or a pair of antheridia are formed in an antheridial chamber inside the thallus. Then archegonia in Anthoceros. So this is a archegonium of the Anthoceros. Then TS sporophyte of Anthoceros. So here's LS sporophyte of Anthoceros. So this will be the diagram that we have to provide in this type of the question. Again VS thallus of Anthoceros. So again here the VS thallus of the Anthoceros, same type of the question. So when you have to see these 5 mark questions or 2.5 mark, you will find that the very commonly the questions are asked on the either internal structure of the thallus or the LS of sporophyte, and rarely the two questions are asked. One is from the antheridium and another that is from the archegonia. Then the 1 mark question on the Anthoceros, the first one, that is the mucilage cavity. We know inside the thallus there is a presence of a mucilage cavity, and this mucilage cavity is filled with a mucilage along with the algal colonies which are involved in the process of the atmospheric nitrogen fixation. Then pseudo elaters. If you see the structure of elaters in case of the Anthoceros, you will find that it is somewhat different than that of the true elaters. and hence it is known as the pseudoelater. They are again hygroscopic and are involved in the dispersal of the spores or release of the spores from the sporangium. Then slime pore. If you see this mucilage cavity, it open to the ventral side or the lower side. It is by means of the pore, and that is known as the slime pore. Then next one more question, that is the involucre. If you see this structure of the sporophyte, you will find that here there is a presence of a extra supporting vegetative cell at the base of the sporophyte, and that is known as the involucre. So this is a cap-like supporting cells which are present at the base of the sporophyte. Then pyrenoid. If you see each cell of the Anthoceros, here along with the nucleus and other, there are the presence of the pyrenoid. The presence of pyrenoid is a characteristic feature of the algae, and if such same type of the pyrenoids are present in the Anthoceros, it means that they show the similarity or the evolution from the algae. Then foot is a part from which or through which this sporophyte is attached to the thallus and is involved in the absorption of the food material during the development of the sporophyte from the thallus. Then columella. If you see the structure of the sporophyte, here there is a presence of a central part in case of the sporophyte of Anthoceros, and this is a central part which is a sterile or is not involved in the formation of the spore, that is known as the columella. The third genus in case of the bryophyte, and that is Funaria. Here the most common question on the Funaria is a sporophyte, as we have seen earlier in case of the bryophytes as well as the Anthoceros. So LS of sporophyte and VS of sporophyte and LS of both are the same. VS, that is the vertical section, LS, that is the longitudinal section, but the meaning of both is same. That is, we have to provide this diagram C. Then gametophyte of Funaria. We have to provide the structure of gametophyte for the 2.5 mark. Earlier you have seen that there is a presence of such type of the plant body which is provided with the structure, that is the stem or the axis, then leaves or the scales, and presence of the rhizoids. So this is a gametophyte of the Funaria. Then LS capsule of the Funaria diagram only, means we have to provide this diagram. Then LS of sporophyte of Funaria diagram only the word is changed but the question is same. Then LS antheridial branch of the Funaria. This is a very different and the different question than that of the other bryophytes, that is the Mertensia and the Anthoceros. That here the question is asked on the antheridial branch as well as the archegonial branch. Then LS of male head of Funaria and antheridial branch of Funaria, male branch of Funaria, all these are the same 4, and we have to provide this diagram along with the structure of antheridia. While in case of the female branch of Funaria, we have to provide this female branch structure along with the structure of archegonia. Then TS stem of the Funaria. This is a diagram showing the TS of stem of the Funaria, where there is a presence of the epidermis, then cortical region, and few cells which are present in the center and provide the support. Then LS male gametophyte in Funaria. The same question, that is the male branch. So we have to provide this structure of the male gametophyte as it is involved in the formation of the antheridium. Then one more questions in case of the Funaria, that is the peristome. If you see this structure of the capsule, then you will find that here there is a presence of a rim of the cells, and this rim of the cells is known as the peristome, which is present to the top of the capsule. and it is made up of the many peristome teeth. Then upper culum is a cap-like structure which is present at the terminal end of the sporophyte, which separates during the release of spores when it dehisces. Then seta is present in between the foot and the capsule, and depending on the length of the seta, the capsule is raised from the foot. Then autoecious means when there is a development of the male reproductive organ and female reproductive organ on the different branches of the same plant, then it is known as the autoecious condition. As we have seen in case of the Funaria, there are the male branch as well as the female branch. On the male branch, there is a development of the antheridia, and on the female branch, there is a development of the archigonia, but on the same plant. So such type of the condition is known as the autoecious condition. Then paraphysis, if you see these structures of the male reproductive organs as well as the female reproductive organ, so here the sterile branches which are present along with the antheridia, that is known as the paraphysis. If you see the same case in case of the female branch or the development of the archigonia, then you will find that here there are the certain sterile leaves which are developing along with the archigonia, and that is known as the perichichial leaves. Then annulus is a group of the cell which is present at the corners of the terminal part of the sporophyte of Funaria and are involved in the dehiscence of the capsule. Then columella again is a central sterile part which is present in the capsule or the sporophyte of the Funaria and is known as the columella. Then apophysis, if you see the certain solan part at the base of the capsule of the Funaria which join the capsule to that of the seta is known as the apophysis. So in this way, different type of 1-mark questions are asked in case of the Funaria. In 4th unit, here there are the mushroom cultivation topic, and in case of the mushroom cultivation, here there is introduction of mushroom cultivation, then different type of the mushrooms, that is edible mushroom, poisonous mushrooms, then method which is used in the development of the mushroom or cultivation of the mushroom, and economic importance of the mushroom. So in case of the 5-mark questions on mushroom cultivation, first one, that is the economic importance of mushrooms. So whenever this question is asked, we have to provide the economic, that is the properties of the mushroom with respect to the food, and second one with respect to the nutrition, along with the medicinal importance of the mushroom. Means here we have to provide the nutritional value of the mushroom as well as the medicinal value of the mushroom in case of the economic importance of mushrooms. Then poisonous mushrooms, though the many varieties or the many species of the mushrooms are edible, there are again many mushrooms which are poisonous also, and we have to provide such type of the poisonous mushroom in this question. Then method of cultivation of Wolveriella wolvacea. We know there are the 3 different species, that is Wolveriella wolvacea, then Agaricus bisporus, and the Pleurotus citrinophyllatus. So these are 3 different mushrooms which are included in the syllabus regarding the edible mushrooms. So we have to provide the method of cultivation of the Wolveriella for 5 marks, or the cultivation of Agaricus bisporus also. Then mushroom tools. We know for the cultivation of the mushrooms there will be the many tools, and that we have to provide in this question. Then the technique of mushroom cultivation. We know during the mushroom cultivation there is need of the spawning, then transfer of the spawning, caching, then fruiting, then harvesting, packaging, storage, All these are the different methods or the technique which are used in the mushroom cultivation. Then infrastructure for the mushroom cultivation means the space and the structures or the thatch house which are used for the mushroom cultivation, that we have to provide in infrastructure for the mushroom cultivation type of the questions. Then preparation of medium and the spawn. So for the cultivation of the mushrooms, there is need of the preparation of the medium as well as the preparation of the spawn, and there is a specific method which is to be used for the medium and the spawn preparation, and that we have to provide in this question. In case of the 2.5 mark questions on mushroom cultivation, again see the importance, economic importance of mushrooms. So same question was asked for the 5 marks also. Again, the medicinal value of edible mushrooms and nutritional value of edible mushroom, and both of these we have to include in the economic importance of mushrooms. Then again poisonous mushroom, that we have seen also in the 5 mark question. Then Wolveriella wolvacea, Agaricus bisporus and Pleurotus citrinopilatus. Wolveriella wolvacea is a padistra mushroom. Agaricus bisporus is a button mushroom, and Pleurotus citrinopilatus is known as golden oyster mushrooms. So these are the 3 different type of the mushroom which are involved in our syllabus or included in our syllabus and can be separately asked for 2.5 marks. Then spawning is a method of the development or the formation of the spawn or the vegetative hyphae of the mushroom for the mushroom cultivation. Then thatched house, these are the huts or the structures which are made of the different materials, very commonly the straw and other matter, and these are used for the cultivation of the mushrooms. Then substrate preparation for the cultivation of the mushroom, there is different type of the substrates, either maybe the padistra or the other any other straw, or even there is a development of the compost. So when we are using that substrate for the growth or cultivation of the mushroom, then we have to use a particular method. Then bed preparation for the padistra mushroom, then bed preparation for the sugarcane trash, and the bed preparation for the banana leaf. So all these 3 different type of the beds are used for the growth or the cultivation of the mushrooms. So such type of the different questions are asked for 2.5 marks in case of mushroom cultivation. Then the 1 mark question that is on the mushroom cultivation is mycophagy. If you see the different condition, that is vegetarian, non-vegetarian, in similar type, if the anyone eating the mushroom, then it is known as the mycophagy. Then phalloidin and amatoxin, these are the components secreted by the poisonous mushrooms, and these are secreted or these are fatal to the human when consumed the poisonous mushrooms. Then padistra mushroom, earlier we have seen that Wolveriella wolvacea, that is known as the padistra mushroom. Then Pleurotus citrinopilatus is also known as the golden oyster mushroom. Then spawning, we have seen that development of the spawn. Spawn is nothing but a vegetative body or the hyphae which are involved or which are used for the cultivation of the mushrooms. Then casing is a method in which the thin layer of soil is applied on the hyphae, and that is known as the casing. Then thatching is a process in which the straw is used for preparation of the roof of the huts or the houses which are used in mushroom cultivation. Then sterilization. During this process of the mushroom cultivation, there is need to maintain the sterile condition so that there may not be any bacterial or the viral diseases on the mushrooms, and hence there is use of the method of sterilization to kill the microbes on the different materials that is used in mushroom cultivation. Then gills, a fruiting body of the mushroom that is provided the vertical structures below it which are involved in formation of the spores or the basidiospores and are known as the gills. Then simply mushrooms are the different structures or the fruiting bodies of the fungi and are involved to provide the food to the human beings. Then compost is specially prepared from the different organic materials for the growth or cultivation of the different type of the button mushrooms. Friends, I hope this lecture will be useful to you in preparation of examination, and also the same type of the questions will be there in university examinations. If you feel that this lecture is useful to you, then kindly like, share and subscribe our YouTube channel Botany for You. And also press bell icon for the alerts of every lecture to be uploaded on different life sciences titles. Thank you very much for watching this lecture completely. Thanks a lot once again.